we can probably go ahead and get started. We'll have a few more people join over the next 5 or 10 minutes, I'm sure. So, all right. So thank you everybody for joining. We'll go ahead and get started here. This is module 4.2.1, guest presentation from Dr. Qiang Zhang from Emory University. He'll be presenting today on a multiscale spatial modeling framework for the germinal center response. This is work that he and a few other people, including previously a high school student, who I guess is in college now, have been presenting for the last couple of years and been working on. So I'm excited to see this year's presentation of it. Just as a reminder, this session is being recorded. We are not currently live streaming, but the session will be uploaded to YouTube over the weekend for other people to view. So please do keep in mind that the session is being recorded. This slide deck can be found in the student materials folder, specifically in the day 4 folder. I will go ahead and grab the link to that, direct link. Sorry, somebody created this folder with editor permissions. Let me update that. Okay, there is a link in the chat. And so if you want to go ahead and download those while Dr. Zhang is doing his introduction, we will have some exercises for you to do in the second half of the talk. And last thing, I do want to call out our funding and support networks here through the NSF, previously the NIH as well. We have support through the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiatives. So I think without further ado, I will go ahead and turn things over to Dr. Zhang to introduce his work to us. So you can
Okay.
share.
Yes. Thank you, Hayden, for the introduction. Does everybody hear me okay? All right. So let me share my slides. Entire screen. Let me do the entire screen thing. Okay. All right. Do you all see the full screen of the PowerPoint presentation?
Yes, looks good.
Right. It's good. Okay. Again, I'd like to thank the workshop committee for inviting me again this year for this demo session over here. I think this is the second time I'm doing this. As Hayden just mentioned, that actually this is the first time we as a group doing this. The first 2 years was done by my student Derek, who's actually right now at Carnegie Mellon University's. Yeah, I think he's probably at his second year in the university now. All right. So I really like to take this opportunity to demonstrate some of the work we have been doing for modeling the germinal center response using the CompuCell3D platform, and also using this demo opportunity to show you guys how actually we can export the CompuCell3D simulation data and then do some post-simulation processing to visualize the simulation results. So here is the learning objective of this session. So I'll first give a brief introduction about the biological ideas behind the germinal center response, which is responsible for generating high-affinity antibodies. And then I'll go over a few, the script about how we actually using CompuCell3D platform to implement this germinal center response model. And the second half of my presentation will be actually, showing you how to export and visualize the data generated by the simulation. And then at the end, you have some opportunities to do some exercise on exporting and visualizing data. So we all know that when we get exposed to some pathogen, like COVID-19, the virus, what have you, right? So essentially, our body will respond to the pathogens by producing antibodies. All right. And this response is what we call adaptive humoral immune response. And what happens is that when we first encounter the antigen or the pathogen, in a few days, our body going to launch a quick response. And in that response, mostly the IgM, kind of what we call a polyclonal IgM antibody going to be produced. All right. And then about a week after that, then the body also produce what do we call isotype switched antibodies. In this case, mostly shown as the blue line over here, which is the IgG. And these IgG type of antibodies is more high affinity. So they have a higher binding affinity for the antigen or the pathogen and be able to neutralize them more efficiently. So as a result, these pathogens going to be annihilated, and then we recover from the infection. Okay? So this is called the primary response because that's the first time you get exposed to the pathogen, and then at some later time when we get exposed to the same pathogen antigen again, then the body going to launch what they call the secondary response. In this case, the IgM response is still similar as the first encounter. However, the second response, which produce IgG will be much higher because of the memory B cells are being produced. Okay, so this is also called a recall response. So this entire process is what we call the adaptive humoral immune response, and is being underlined primarily by the B lymphocytes. So the B lymphocytes in our body is actually produced in the bone marrow by the hematopoietic stem cells. And they go through a number of intermediate steps, all right? And then the naive B cells will eventually move out of bone marrows and then go into what they call the secondary immune organs, including lymph nodes and also spleen, where they will be resting over there to the immunosurveillance as mature naive B cells. So when the body encounters antigens, these antigens going to be able to stimulate the naive B cells and to stimulate them become plasma cells. So the plasma cells are the terminal differentiated states of B cells. And those are the cell types that secrete the antibodies, either IgM or IgG or other type of antibodies. So in terms of the IgM response and the IgG response I mentioned early on, that is actually being mediated by two different responses. One is called T cell independent response, which means a subset of B cells will be able to respond to what we call polyclonal antigens of the pathogens, such as lipopolysaccharides and some DNA fragments of pathogens. And as a result, these B cells will be transiently activated and then eventually terminally differentiate into the short-lived plasmablasts or plasma cells. And those ones that's actually secreting the IgM antibodies you see early on. Okay, and this happening within probably 3 or 5 days after the infection happens. However, these IgM, they're usually kind of low or intermediate affinity. So their kind of effect in terms of neutralizing the pathogen antigen is kind of not ideal. So what happens with the body is that they also launch what we call a T cell dependent response. In this case, if the follicular B cells in the lymph nodes or the spleen will get activated. And these cells will actually proliferate a number of times and through this what they call the germinal center response. And as a result of this germinal center response, it will produce what we call the long-lived plasma cells. So these plasma cells, they can live almost forever, and they move eventually back into the bone marrow and to stay there. And so these plasma cells produce antibodies to provide lifelong protections against the same pathogen. Also in the process, some memory B cells will be also produced, which is also long-lived, and they can be quickly recalled when our body encounters the same pathogen again, and to produce the secondary amplified responses. Okay. So this T cell dependent response is really responsible for generating lifelong high affinity antibodies. So this germinal center response is happening primarily in the secondary immuno organs, including the lymph node, the spleen, and also in some cases like the Prius patch in this in the GI tract. So here, just to give you an idea of the what a germinal center look like. For example, what I'm showing you is a lymph node. The lymph node itself is about like a few millimeters in terms of the size, and then the germinal center, which is forming during a active infection, have a size about probably 200 to 300 microns. All right. And when you look at if you do a cross-section of the lymph nodes and observe using the HE staining and observe under light microscope, you're going to be seeing that the germinal center can be roughly divided into two zones. Okay, the dark zone and the light zone. So the dark zone contains densely packed cells that are actively proliferating or dividing, and the light zones contain cells are actually not dividing. So the germinal center is actually is underpinned by a kind of a cellular dynamics that represents a remarkable spatial-temporal phenomenon. So here I just give you an idea of what's really going on in the germinal center. So in the germinal center, what happens initially is that the B cells that first get activated when they encounter the antigen, they will move into the dark zone area of the germinal center, where they proliferate a number of times. All right, and then they stops. So then these B cells will be able to move from the migrate through chemotaxis from the dark zone to the light zone, where we call these cells as the centrocytes. So these B cells in the light zone will be able to interact with two different cell types. One is called follicular dendritic cells, FDC cells. These FDC cells contains the antigen. They are what they call antigen presenting cells. All right. So on the cell surface of FDC, they having the antigens actually on the cell surface. So the B cells in the light zone on their surface, they're expressing what they call the B cell receptors, BCR. So the BCR is actually a surface expressed IgM molecules. So once these B cells is able to contact with FDC cells, using the BCR, they're able to recognize the antigens being presented on the surface of FDC. And as a result, this B cell will be able to engulf or internalize these antigens and then process these antigens internally. And after processing, these antigens will be chopped up into small pieces called epitope. And this epitope will be re-expressed on the B cell surface with the help of what it calls the MHC2, the molecules on the B cells. Okay, as you see over here. So these B cells, when they're expressing these epitope on their surface, the next thing they can do is to be able to interact with the follicular helper T cells. We call it TFH here. Okay, so these the epitope expressed on the surface of the B cells will be able to recognize by the T cell receptors, and as a result, to activate these T helper cells. So the active T helper cells start to express the CD40 ligands on their surface and also secrete a bunch of interleukin cytokines. And these factors, both the diffusive form and also the CD40 ligands, will be able to activate the B cells. And how much the B cell will be activated depending on how much the B cell activate the T cell itself through this MHC2 epitope and TCR interactions. right? So, and so the more the B cell get activated, the less likely they will die. So as a result, those surviving B cells will be able to migrate back into the dark zone. Okay, and in their dark zone, they will start to proliferate or divide for 2 or 3 times. And the number of times they're dividing really depending on the strength they're being, the degree to which they're being stimulated by the T cells. Okay, and during this is called clonal expansion. Right. So during this 2 or 3 or 4 time cell division, these B cells will start to go through what they call a somatic hypermutation, which means the genes sequence encoding the heavy chain of the immunoglobulin will be mutated through point mutation for every single cell division. So as a result, once a mutation happens, the antibody affinities that's being encoded by this heavy chain can either improve or get worse, right? Because mutation are always random. Right? So sometimes if the mutation is actually causing the formation problem of the antibodies, then those cells will also go through apoptosis. Okay, but if the mutation is kind of benign, that means it's only vary the affinity of the antibodies, nothing else. Then these B cells, after they stop proliferating, they will migrate back to the lysosome again. Okay, so with the antibody or the BCR affinity for the antigen changed from the previous cycles. So these, as I said, this affinity of the BCR can be either improved or getting worse. So, but they go through the same selection process. Only those have high affinity for the antigen will be more likely being selected by the T cell over here, and then moving back to the dark zone and proliferates for a number of time again. Okay, so this is essentially a kind of Darwinian process, and as a result, only those B cells that happen to mutate into antibody sequence that have high affinity for the antigen will eventually survive. And this is what's going on with the germinal center. So as you see, this is really kind of a spatial temporal phenomenon, very remarkable, and happening probably in the first week of the first antigen encounter and lasts about another 2 weeks. All right. And the force that's driving the migration between dark zone and the light zone, the B cells, is driven by these chemokines, what they call the CXCL12 and the CXCL13 over here. Okay, so essentially the CRC cells in the dark zone, they're secreting the CXCL12, all right, establishing gradients have a high concentration on the dark zone and low concentration in the light zone. And then the follicular dendritic cells secrete CXCL13 and establish a opposite polarity in terms of the gradient. Okay. Then the B cell will alternately expressing the receptors for these two chemokines. One the CXCR4 for CXCL12 and the other CXCR5 for CXCL13. So as a result, because these cells kind of alternating expressing these two receptors, that's causing these cells to be able to cycle between these zones. Okay? So essentially, what happens in the dark zone includes clonal expansion, somatic hypermutation, and apoptosis. And in the light zone, what happens includes positive selection, apoptosis, and the plasma cell formation. Okay, so here just shows you the cover page of the paper we published. We're trying to model that germinal center dynamics and using the CompuCell3D through this kind of multi-scale spatial modeling framework. Okay. And so here, because it's a multi-scale, that means we're combining the molecular network, using molecular network to drive cellular behavior, and that in turn will lead to formation of the germinal center at the tissue level. So you can think about this as a molecular level, cellular level, and tissue or organ level kind of a cross-scale simulation. The genetic network that's really responding to the stimuli, including the antigen stimulation through BCR and CD40 ligands through CD40, are really complicated. So the wiring diagram I'm showing over here is just a partial understanding of what's going on in the real B cells. All right. So for the purpose of our paper and also for demonstration, we are able to kind of simplify that complex network into a more streamlined, readable format, as you see over here. All right. So there's a bunch of the signaling proteins as well as transcription factors are driving the process. And some are responsible for driving the cell growth, cell cycle, and some are actually driving the somatic hypomutation, or apoptosis, and some are responsible for driving the migration as well as terminal differentiation into the plasma cells. All right, so here, when we do a model, all right, we try to keep the model as simple as possible and sufficiently complex so it serves our purpose. For this particular model, we're capturing these key biological events. So we're in the model, we're including these major participating cell types: the B cells, follicular dendritic cells in the lysosome, and the CXCL12 expressing reticular cells or CRC cells in the dark zone, and the T helper cells in the lysosome. And then for the B cell chemotaxis, we're using the chemoattractant ingredient you see early on to drive the migration. And for the lysosome B cell interaction and outcomes, we actually simulate the interaction of the contact of B cell with DCs, and the result of that interactions, we're having the BCR mediated surface expression of the MHC2 complex and also the signaling induced by that complex. And in terms of with interaction with the T helper T cells, in this case we're modeling CD40 mediated signaling, which cause positive selection of the B cells that have a high antibody affinity, and also driving them into this dark zone reentry. And also the lysosome, we have this default apoptosis or death timer set up. And in the dark zone B cells, we're primarily describing the B cell proliferative burst, which we also call a clonal expansion, and also somatic mutation, as well as apoptosis if the mutation deemed to be too damaging. Okay, so here is actually a recording of the simulation, and in this case, this is from the published paper here. So we're trying to simulate. We're starting, I think, with like 200 B cells, and in the middle of the germinal center, and then we'll see that the color of the B cells will actually represents the antibody affinity, right? So. So here's the simulation. As you will see that at the beginning, these cells will be migrating between the light zone and dark zone, all right? And multiple cells will disappear because of apoptosis, because at the beginning, their affinity is either low or intermediate. But over time, as you see that they're migrating through these two zones, the color of these cells become kind of greener and then become yellower and they eventually become red. And also the number of B cells will become more and more. That's because they're going through this kind of Darwinian selection process. So only those B cells that have high antibody affinity will be positively selected, and then those will proliferate, and eventually the full germinal center will be filled with B cells. All right. So I'm just quickly advancing this process. As you see, that's the B cell color will become more greenish, yellowish, and eventually some of them actually have a red color representing very high affinity. So eventually this whole space going to be filled by high affinity B cells, which either red or yellow or gray, right? So this gives you the visual of what's going on with the simulation that we published in this paper. Okay. So the next slides. Oh, okay. So no, actually, yeah. The next slides actually give you some snapshot of doing that whole process. In this simulation, 100 MCS or Monte Carlo steps is about equal to 1 hour. So for the whole process, we're modeling about a few weeks essentially. And okay, so here shows you how the affinity maturation occurs. So for this simulation, we're starting all these B cells with an intermediate affinity level, about 5, right? So they are starting at 5, but over time, at the somatic hypermutation, some B cell will actually start to have a higher affinity, and some other B cells will have a lower affinity. So you can see the affinity will start to diverge, all right? But because of a positive selection process, the mean affinity over time progressively drifting to a higher level, all right? And we have a wider dispersion of distribution of affinity in these B cells. But only when those B cells have a very high affinity, that's going to trigger this terminal differentiation. Those are the B cells, the green dot here, that eventually become the plasma cells that secrete high affinity antibodies, right? And also during this process, you see that the number of B cells in the entire germinal center also gradually increase. And the dark zone and light zone roughly maintain a kind of a 2 to 1 ratios, which is kind of consistent with what has been shown in the literature. All right. So that's give you the idea of what is germinal center and how actually we simulate that. So for the demo purpose here, we're not actually giving the full CompuCell3D script because it's really complicated and very slow. And it probably taken overnight or 24 hours to do a complete simulation. So here we're giving some simple examples, but the main idea is still there. So in terms of the CompuCell3D model itself, as you have learned through the last few days, essentially contains a main, a Python script, and then with some XML script, and the Python script defining all these functions, and the pip file defining the position of the cells. And so by using this script, we'll be able to produce the simulated data, and that simulated data will be saved, and then will be processed by a Python script outside of the CompuCell3D, and then be able to plot the simulated results, right? And those the outside of CompuCell3D, we call it main analysis script. Okay, so here, just okay for this demo purpose, we're only doing a 2D, so we're kind of making this as 100 by 100 pic by 1 kind of a 2D pixel space as defined in the XML files over here. In the actual simulation we did for the paper, we're actually expanding that to 250 by 200 by 11 at the 3D space. And also in this exercise, we're defining a rectangular wall. Actually, no, it's a kind of a square wall. For this particular purpose, but actually for the model we did in the paper, we find out that we really don't need the wall because all the chemoattractant factors, attraction force will be able to gravitate the B cells. So there's no B cell actually leaving the simulation space. So we don't really need that wall. All right, so here the seeding. Okay, so then we're here showed you how we actually seeding these simulations with initial cells. And for the CRC cells in the dark zone, we're doing this by via a random ellipse-based generation. And then the FDC and the T cells in the light zone were generated in the light or similarly. So essentially, we're just drawing random numbers and follow the equations for the ellipse, and thus be able to produce these randomly positioned CRC cells in the dark zone and the FDC and T cells in the light zone. However, in the published paper, we position these cells in predefined locations for the purpose of model reproducibility. Because if you have a lot of random generated cells positions, then the results of every simulation will become different, and you have to do a number of those to get some statistic consistent results, right? And then, okay, so here just give you an idea. That's for the demo purpose. We're generating these rectangular CRC cells in the dark zone here randomly. And then we're generating these small T helper cells in the light zone and these rectangular FDC cells. And as a matter of fact, the follicular dendritic cells, they have long dendrites. That means that they're more like star-like, okay? So at this stage of our model, we haven't been able to do that yet. In the future iteration, we may be doing that, right? And then for the published results, so what we did is actually using the, as I said, have predefined CRC and have predefined positions of the FDC as well as the T helper cells you see over here. Right? All right. So the next thing you want to do is that you want to assign an antigen sequence to the FDC cells. Remember, the FDC cells are the antigen presenting cells, right? So they're holding those antigens on their cell surface. Okay. So here we're using the four nucleotides, AG, right, TC, to represent that antigen sequence. But we're doing this just for convenience because remember, the antigens, they're essentially most of them are proteins, right? So they're supposed to have amino acids to form their sequence, all right? Not nucleotides for DNA or RNA. But here we're just using these ATCG four letters for convenience to represent that a particular antigen has a particular ATCG sequence. So we're for simulation, we just predefine, for example, this particular antigen will have a sequence of AGTCT. Okay, that's one we predefined. And then the next thing you want to do is to establish the B cell clones that seed the germinal center. And again, we want to, because each B cell clones will have a antigen sequence that's slightly different from each other, and also which means their affinity for that antigen sequence will be different. All right. So here we're just starting these B cells with random sequence by drawing random numbers from this ACGT alphabet. All right, and then randomly assigns to each seeder B cells to begin the simulation. Seeder B cells were generated with random location and assign a sequence that says containing these randomly generated antibody sequence. Okay. And then the next thing is when a B cell is being born, right? You want to initialize them with a target volume and also the lambda volume parameters over here. And then because these B cells need to respond to these chemoattractant CXCL12, CXCL13, so you also assign these chemotaxis using chemotaxis force, using the chemotaxis plugin to these cells once they're being generated. And then the lambda value really set up how fast they're responding or moving towards that particular chemoattractant. And then also at the end, you want to assign this B cell network, the gene network I mentioned previously, also to the B cells that are being born. and we're using this the Gillespie stochastic simulation algorithm to model to simulate the gene network behavior. The chemotaxis force here is being defined in the XML file, as you see over here, the CXL13 and then also the CXL12. And in addition to the diffusion constant, decay constant, and we're saying that it's the FDC cells that are secreting CXL13 and the CRC cells that secrete or produce CXL12. Okay. And then, okay, so here give you the idea that the chemotaxis gradient you'll be seeing with the model we published in the paper. So because the CRC cells produce CXL12, so you have a high concentration on in the dark zone and the low concentration in the light in the light zone, and CXL13 has a inverse polarity. Over here. So, okay, so here is the kind of a Tellurium models for the intracellular molecular network. So these network are responding to these chemokines, as you see here, and also they're responding to the T cell stimulations to the CD4D ligands, as well as responding to the antigens that are being presented to the BCR. And that in turn triggers a number of intermediate signal transduction steps, eventually causing upregulation or downregulation of these intermediate transcription factors. And each one of those is able to drive certain cellular behavior, as you see over here. So because of the time, so I would not go over in details of these steps over here. Okay. And this is Tellurium gene network model in that particular format. Okay, so here, okay, how do we model the B cell interaction with the follicular dendritic cell and T cells? Okay, so here we're using the neighbor checker plugin to determine whether or not the B cell is in contact with an FDC or T cells. So in the XML file, we define this plugin over here, and then in the Python main script, essentially, what you're doing is that you first examine the neighbors, right? You get cell neighbors, and then put that neighbors into the list, all right? And then you go through the neighbors, all right, by type. And then because in our model, the FDC cell type ID is 2, so we're saying that if the neighbor contains a cell which has an ID of 2, that means this B cell is in contact with FDC. So this variable touch FDC become true. And once that's become true, that will trigger a number of events. And so, for example, once this variable becomes true, that means it's interacting with FDCs, then there's a few things happens. The first thing is because the B cell will be using the BCR to recognize or bind to the antigens. The first thing is it obtain the antigen sequence. So it will be able to read the antigen sequence of that that's presented by that FDC cells, all right? And then that antigen sequence will be compared with the antibody sequence that's contained or hosted by that B cells, all right, to obtain the affinity score for the antibodies that particular B cell is actually producing. Okay, so it's comparing just the letter by letter. If they're all matching, in this case, we're giving a high affinity score of 5. If they're no matching, that will be 0, for example. And then with that obtained affinity score, then the next thing is the model we'll be doing is checking whether the B cell will be in contacting with the T cells. And and depending on that particular score, that will in turn determine the probability the cell will survive, which means we'll turn on the NF-kappa B signaling pathways. All right, we're assigning the production term of K7 into 10. So as a result, the NF-kappa B will become activated. All right, and then that in turn will also trigger the expression of the CXCR4, which will be responding to the chemotaxis CXCL12 that's being expressed in the dark zone. All right. So as a result, if by chance, okay, depending on the affinity score, okay, if by chance, where's the random sampling here? Okay, here the random sampling. So we draw a random number and compare that with the percentage chance determined by the affinity score. Okay, so if the affinity score is high, then the chance of the cell survival will be high. Then more than likely, which means those are the cells that tend to have a higher affinity. Those are more likely to survive and then express CXCR4, and then will migrate back into the dark zone to proliferate a number more times. Okay, and in the meantime, it will turn off this death timer, so those cells will be surviving. Otherwise, if these cells are not selected by this random number comparison, then nothing will happen. So the cell have a default death timer. They will just die at some point after some time elapse. All right, without returning to the dark zone. Okay. Oh yeah. So here is the term is kind of the how the cell death is being implemented. So essentially, we're saying that if the NF-kappa B level is low, like say in this case below 50, then we're assigning a non-zero value for K13, which starts the death timer. All right. So initially the death timer is at set 1. Once it becomes 0, and then the cell will be deleted from the simulation. So that's how the cell apoptosis occurs. And okay, so for those B cells that survive and also migrating back to the dark zone, and then we're checking whether AP4, which is downstream of NF-kappa B, and also the MYC, those are the ones driving the cell proliferation. Okay, so we're saying that if AP4 level is high enough, that's going to trigger cell growth. So the volume, the cell volume will be growing with a kind of exponentially, and that how fast it's growing is actually depending on the doubling time. In this case, the B cell doubling time, at least for mice, is about probably 5 hours. Every 5 hours they double, all right, the number of B cells, and so that's kind of the self, and so the cell proliferation is supported by AP4 level above a certain threshold. However, if the AP4 level drop below threshold, then the whole activation pathway is being turned off, and then the CXCR4 expression will be turned off, and then the lambda value for responding to the chemotaxis CXCL13, which expressed in the light zone, will be turned on. So as a result, these cells will be leaving the dark zone and going back to the light zone. Okay. Oh, then here, so it's about the script about cell B cell division or mitosis. So essentially, we're saying that if the target volume hits a threshold, in this case 32 pixel volume, and then the cell will be will spawn a daughter cells, and then the daughter cell will inherit everything of the parent cells, but then all the molecules will be actually the molecule numbers in those gene network will be halved or equally divided between the two daughter cells. All right, but every other things are being inherited. For example, the affinity score, the XY coordinates, etc. Okay, so and also during this process, where we have to do this random hypersomatic hypermutation. So what we did is that we'll pick a. Remember, in this case, we're using the 5 letters represents a antibody or antigen sequence. So for the B cell antibodies, we're just among that 5 letter sequence, we pick a random position, and then we remove the letters and then replace with a randomly selected letters to represent this somatic hypermutation. So this script actually implements that idea. Right. So as the simulation going, we try to save the results of the simulation at some interval. In this case, we're saving this into a TXT files for every 15 MCS, right? So this is script doing that. And the save frequency, which is 15. And then, so that means the Monte Carlo MCS, when it hits the multiples of 15, it will save the data. So the data and the naming of the data file is as follows, where A represents the generation where the B cell is. We're starting with generation 0, and letter B represents the mother ID, and this letter C, I mean the position C represents the actual cell ID. Okay, so here give you the idea that these for each text file generated represents a cell. All right. So, like for example, the number 1 here means that the first generation and the mother ID is 59, and then the cell ID of this particular cell is 64. Okay, and like the mother 59 produced 2 daughter cells, 64, 65. Yeah, so it just so on and so forth. And here gives you the idea what's in those text files. Essentially, it has a header, and then the first column has represents the MCS every 15 MCS over here, and then the next column gives the cell ID, and then there's all the parameters are being recorded, so like the mother ID are being repeated, 59 over here, and then here the remaining of those actually recording the variable levels like MCH antigens, the lambda values for the chemotaxis, and then the variable values, for all those genes in the gene network. Okay, so this goes on until the cell either die or differentiates into the germinal, into the plasma cell, or the simulation is being terminated. All right. So I think this is the points where maybe we can just quickly show a demo of this simple model. All right. Okay, I see there are questions. I'll save the questions to the last, all right, and answer them. All right, so here you see the GCR demo, right? This is the main one here. Nothing unusual, basically importing all these steppables that's being defined in this GCR demo steppables over here. I already show you some of the scripts, so I wouldn't go over all of them. And then you have the pip file defining, in this case, at the wall here, and then the XML file. All right. Okay. So when you do your simulation, one thing you probably need to change is the the directories where these TXT file being stored. So in my case, I'm saving this to my local desktop. So that's the script I have over here. So I generated a cell data folder in my desktop over here, as you see. All right. So nothing over here is empty. So if you're using Windows, it's very confusing because there's two desktops. One is local to your user, and if you're using OneDrive, and there's another desktop actually, the desktop of the OneDrive shared drive. So sometimes can be confusing. So yeah, so you can just simulate this. I usually right click here, say open in player, and that will start the simulation. Okay. So as you see here, in this case, the color shows I think in this case, the MIC level. But let's show something like the affinity, or let's see, or the affinity score. Okay. Yeah. So these cells, okay, these square cells, stationary square cells are the CRC, and the stationary square cells are, remember, are the FDC, and the small are the B helper cells, right? So the one moving are the B cells, and they will be changing colors as they're going through proliferation and mutation. And then some will start moving back to the light zone. So this, yeah, we won't be able to complete the whole simulation because it's kind of slow, right? But in the meantime, you see that a number of textile is being generated over here. All right, and these are I think I'm starting with 6 cedar B cells, so that's why you have these. And then if you open one, we have all these things as I showed you in one of the previous slides. All right, all these variables are all over here. So basically, I'm recording everything at least in the gene network, and some other related parameters: cell generation, number of mutation, yeah, cell volume, precision, everything is all in there. So these saved similar data allow you to do post-simulation visualization and also analysis, right? All right, so I'll just let these run. And then now I go back to my presentation. Okay, so here, right, I want to just go through this kind of the main Python file for post-processing, all right? So yes, I showed you this text file in the cell data. And then in that main Python file, which actually I use Spider, all right? So this is the main Python file, which actually in your folder is underneath this directory, simulation result analysis. That's the main analysis script, .py. All right. And then these cell data are the one I pre-saved. It's not the one that's being generated right now. Okay. So once you open this in Spider, I don't know what's platform you're using. Make sure your current directory is also in it's the same at this main script, right? In Spider, it's straightforward. You can just right click on the tab, say set console working directory. That will automatically set your directory into the same directory as where this main file is being stored. Okay. Okay. Now in that script, we have defined a function that's to read these txt files line by line. All right. So that's what this function is doing. And after that, we also define a function to recursively find the lineage. right? So lineage is defined as you starting with a seeder B cells, right? Every daughter cells and the daughter cells of daughter cells are being generated. That's branching tree of the lineage, right? So so here essentially we have a function to basically traverse that binary tree, be able to establish all the daughter cells, and that comes after that. Yeah. Um, and so essentially, for each seeding B cells, we do this lineage branch retrieval process. So the idea is that, let's say you're starting with a seeder B cell number 62, right? And you generate all these binary trees, right? And then if you follow any one linear path, that's give you a lineage, a branch, right? If you're going through 84, 93, let's give you another linear branch, give you another linear branch, right? And then for the 63, you do the something similar, right? So the terminal cells in a linear branch represent either cells that die or B cell that's differentiated into plasma cells or is at the end of the simulation. That's the last cells in a branch. And so when you execute those lines, it will produce all the sequence of each lineage. Like say this one starting from this, ending over here, and this will also starting the same cell but ending with a different cell, and so on and so forth.
Whoops, sorry about that.
Did I stop? Okay.
Yeah,
No
my fault. I
problem.
clicked on something and
Okay.
there we go. Okay.
Right. Okay. Uh, and then the next thing that's what you can do is you just pick a lineage you think you're interested, and then do further analysis for that single lineage branch. Oops. Hold on. All right. Yeah. So once you pick a branch, the next thing that's being done is to combine all the cells in that lineage. Essentially, you're appending all the TXT files of the B cells in that lineage branch. And that happens in a temporal order because, right, later cells are being generated at a later time. So you can just append them all together to generate a huge kind of a matrix. And then the next lines over here will actually ask you, to ask a user to say, okay, what variables you want to visualize or display. So essentially here are the kind of input interface for that. So that's basically, then at the end of the script, it's about how you visualize, you do some data type conversion because the file, the TXT file, those are being stored as string, right? So they need to be converted into floats or other data type, array in this case, and which allow you to do the plotting. And these are the 2D or 3D plots, which I'll show you. Okay, so here give you the idea that after using those the analysis script in the paper we published, 3 lineage branches ending with different fate. For the one on the left, it starts over here, but after going through a number of times, so the color represents actually the time, all right? Then these cells actually die over here and in the dark zone as a result of what we call mutational death. And these cells, starting over here, after going through a number of migration back and forth, is die over here in the light zone because it's now being selected, positively selected. And this cell on the right, actually starting over here, ending over here in the lysome, and that actually become a plasma cells because its affinity become very high. So it triggered that terminal differentiation. And also with those postscript analysis, you can plot kind of the temporal dynamics where okay, so I don't know why it's not showing. So the x-axis here represents the MCS, okay, the time, the simulation time. You can see, for example, the cell volume, they up and down. That's representing the cell. These cells are actually proliferating for 3 or 4 times in the dark zone, and then moving back to the light zone, not proliferating, and then going back to the dark zone, proliferating for more time again. Yeah, so each time the number of time division varies. And for that branch, you see the affinity, because this is the branch that I picked that end up with becoming plasma cells. So that affinity for that lineage branch kind of, stepping, kind of getting higher and higher, although at sometimes they may be lower, but eventually getting higher and higher. Yeah. And then the CXCR4, which being alternating express or turning off, driving the cell migrating between the dark zone, light zone, what have you, right? Okay, and then so this is results that's we also did to reproduce the spatial variable levels. So these days, people talk about spatial transcriptomics, spatial whatever, right? At cellular resolution. So this is able to give you the idea of that in the formed germinal center, what a particular gene expression level is, depending on the position, the cell position location in the germinal center. So this kind of gives you that idea, and either doing a spatial transcriptomic or maybe a immunohistochemistry staining kind of studies, right? And then here you can also analyze data to show them at these violin plots, trying to give the idea of if you do a single cell RNA sequencing, that's how the dark zone, light zone expression level differ for these genes. Yeah. All right, so this slides give you the idea of, as I showed you some of the earlier ones, the affinity maturation, the growing, and then the cell number growing. And also here, the BC panel give you the number of plasma cells that's being generated after a certain period of time. There's a more plasma cell being generated over here. And then this EF, what we call the Muller plot, just give you an idea that if you're starting with let's say 100 or 200 number of B cell clones, only a few eventually survive. That's called a clonal dominance. That means you start with a number of B cell clones, only a few eventually wins out. The rest of them are being sacrificed or killed during that positive selection process. And that has been shown experimentally as well. Okay. So now let's go back to our simulation here. Okay, so here you see that now we're starting with 6 or 5 B cells, and now there are more B cells are being already being generated, and some of the colors is moving up to this area, indicating their affinity level become higher. All right, compared with the level they start with. Okay. So now let's just go back to the Python code. I'm using the pre-saved data, all right, to give you the idea of to show you to do some demo over here. So what I'm gonna do, I'm just run. Okay, let me hide this stuff. Hide floating. I'm gonna run the first few lines for the library import, as well as the definition of reading files. And so the next few lines is really reading the TXT file in the cell data. folders, right? So if I execute those, you see that's going to produce all these um, file names, essentially. All right. And in the variable explorer, you're going to have all these stuff here. Basically, the files that's reading the file names, right, which represent the cell. And then the next thing is find out how many cells are in there. So in this case, we have 260 cells, right? And then the next thing we do is to read the header files to give you the idea of what contains variables is contained. So those ones are being stored or in the header variables, right? And all right, so the next thing is doing the finding to find the CDR cells, which is the generation 0 cells. So essentially, that what's doing is just to read the file name. If the first letter or first digit is 0, that means those will be the CDR cells. So if you execute these lines, that will give you the CDR cells. See, these are started with 0. So these 5 cells, they're all the CDR cells. Function to retrieve lineage. Right. So this function, as it says, is basically doing the binary tree traversing to retrieve all these lineage and their branches. So you can execute this whole defined function over here, right? That's defined. And then the next few lines, okay, is trying to see whether you really want to call that function. So here, we're having an input yes, because we only have a few hundred cells here. When you have a few thousands, this process can be lasting a few, like an hour or 30 minutes, if I remember. So you don't want to accidentally execute those lines and making the process just hang in there. So we says, okay, you want to ask, do you want to do this or not? All right. So once you do that, you say yes, it will go through the CDR cells, and then the call the retrieve lineage branch function to basically retrieve all the lineage. And then here is displaying that. Okay. So let's just execute all these lines. It says yes. All right. I think it takes probably a minute or something. Oh, actually, faster than I thought. Okay. Now it retrieves all these lineage branches and being displayed over here. And that is being stored in this called lineages, actually, variables here. See here, right? So let's say the lineage, this one started from this CDR cells, ending with this, right? So as going on to further, there are more cells. It's kind of a deeper branches, lineage branches, right? So that's give you all the lists for each lineage branch. And then among these, you can pick one, depending on what you see in the data, to further analyze that particular branch. So for the demo purpose, I'm picking this branch. So I'm just assigning this sequence to this variable. And execute that. Okay. Now the next thing it's doing is combining all these cells in that lineage into a bigger branch data, right? So we're starting with something empty and keep appending essentially. So we do this. So now if you like the branch data, all right. So that one basically contains all these headers, and then these MCS is going all the way down to whatever. Okay, periodically you have these headers being repeated, but representing that starting of the daughter cell of the preceding cells. All right, and you're moving down, you have another header. So eventually, these going to be removed from this list. All right, so here just to display the header to give you an idea what's in there. And then the next thing you want to do is to, want to select variables to display or plot, right? So here, we're just asking, what do you want to display? For example, I want to display the x coordinate. That will be the x com in this one. And the y variable will be the y com. And then for the z variable, it can be anything. So let's say, what about if we do something about maybe the CXCR4, which is the receptor for the chemo CXCR4 for the chemotaxis, all right? So you do this. Now you're saying I have all these three variables, and the next few lines is further processing for data selection, and also like some string to integral conversion, and then the 2D plot. So if you do this, it will plot this. Okay. So you see that's the XY represents that's the actual simulation space of X and the Y coordinates. And so the B cell, I think in this case, starting from here, just keeping going back and forth and forth. And do not process the CXCR4 represents by the color of the color map is being actually is being varied. It's being varied. All right. And driving I've driven the B cells towards. So once the CXCR4 is highly expressed, it's going this way. When it's being terminated, the cell going back to the lysosome. All right. And you can also visualize this in a 3D pattern, the X, Y coordinates. I don't know why it's not showing. Then in the Z direction is the CXCR4 levels, right? So let's do one more thing. Maybe we can show the temporal stuff. Let's try cell volume stuff. So the cell volume is, do I have that here? Score, touch. Oh, I don't have that here somehow. I don't know why. Okay, so what else I can do? Okay. Let's see if I can do a time course thing. Right? Okay, let me do this. 2D skewed. So the x-axis will be the MCS on a color step. Then the y-axis, let's do the SCOM for this one. So you know it's going back and forth. And then the y can be anything. I mean, the z can be anything. In this case, maybe do the y-com. I don't know what's mean. All right. All right, see this? Now let's give you the idea as time goes by, how the cell actually moving back and forth in the x direction. All right, in the x direction. Yeah. And then the color represents where the y direction is. All right. So you can do variable combination of all this stuff. Okay. So the Python script I'm showing here are basically ends over here. But for the more complicated analysis, I'm showing over here all these statistics and all spatial So these more advanced, those scripts are, if you're really interested, you can go to the paper and then just go to the GitHub repository we're showing in the paper to download those to study those scripts, how we do the analysis, right? So with that, I think let me just quickly recap, and then we can answer some questions, right? So essentially, the CompuCell3D model we're doing quantitatively recapitulate the key morphological and functional events of the GCR. And a variety of post-simulation analysis can be performed with relevant variables at single-cell resolution saved at a fixed frequency. And of course, this model is far from being ideal, and there's a number of things that actually we're planning to do to better reflect the true biology of the germinal center. And with that, I'd like to thank Derek. He was really amazing. He did this project while he was like in the last 2 years in his high school time. And then now he's at Carnegie Mellon studying computer science. And also Chris Sherrill and also Norm Kaminsky's, which are my collaborators. And this particular work is being funded by the NIEHS Superfund project, which studies how environmental toxicants like dioxin actually suppress the immune response. And so I think that's all. Thank you very much for your attention. And so let's see what kind of questions you guys are having. Yeah. Oh, actually, I'm not showing my last few slides, right? Where? Okay, oh, here. I think I got lost. Oh, what? I think. Okay, now let's see. What questions are there? So, yeah, the first question, what do the color of the arrow mean? Long arrows, right? So, which slide is that one?
That was on your network diagram slide towards the beginning.
Okay. Am I still sharing?
Yes. Yeah, we can see.
Maybe this one here, right?
Yes.
So the long arrow. Okay. So the long arrow, you mean these blue ones, right? Or these ones, the yellow ones?
Oh, Van is no longer in the meeting. She must have gone to lunch.
Okay, but I can explain both. So the long ones are representing some, like in this case, is the how the somatic hybrid mutation or class switching affecting the BCR, because essentially, remember in BCR, the BCR is actually the IgM antibody that's being expressed on the surface of the B cells as opposed to being secreted. Okay, so the somatic mutation and the class switching will be affecting these BCR, all right, activity or affinity. And if he's asking about these arrows, that's it's just indicating that this is really representing the complexity, and we're simplifying that, and then this is the implementation. Okay. In Tellurium. What's the next question? Is there a reason why 3 space was used in the 2? Okay, so that's a good question. At the beginning, we're starting with the 2D, but what happens is if you have a cross traffic, right, of the B cell moving from the dark zone to the light zone and from light zone to the dark zone, once you have a lot of cells, you have this crowding effect. So the cell is hard to move from one side to the other side because it all jammed up. If you start to have a 2D to give you additional layers, so those cells will be able to, if I cannot cross in that layer, I can move to the other layer to cross, all right, from the one side to the other side. So that's why we're using the 3D in the 2024 paper. And because of actually we're using 11 layers for that Z direction, but actually for the germinal center, it probably goes up to 50 layers, about 50 micron in terms of thickness. So, but if we do that, the simulation will be way too slow. So that's why we kind of just using 11 layers for that paper. So I hope I answer your questions. The next question is, it seems we have some bug in the shared demo file. That's I think I explained that, right? You need to kind of adapt that to your own file directories to be able to save those TXT files. So that's hidden again, um, hidden again. So Osman, did you do validation of your model with actual experimental data? That's a very good question. So in that paper, we actually list a number of things that's being produced quantitatively by our model and compared with the experimental results. So to give you some idea of what I mean, it's including things like, where is it? Uh, uh, for example, the gene expression patterns in the dark zone light zones. So we're comparing our results with experimental data. And we're also comparing. So these things you see over here, in terms of variability as well as the B cell numbers, the ratio between dark zone, light zone. So these are conforming to the experimental data essentially. So the model is actually being constrained by a lot of published experimental data, actually mouse data. Where the question part over here. Did you and if yes, how did you do it? With which experiment? With which experiment? Yeah, I just mentioned a few. There are a number of other experimental data, including I just mentioned the size, the dimension of the germinal center, the number of B cells that's seeding the germinal centers, and then the fraction of the cells are dying in the dark zone, the fraction of the cell dying in the light zone. All those numbers actually were carefully using to make sure our model actually largely conform with experimental data. Yeah, I think I answered all these questions listed in the chat. There are other questions from the audience?
Yeah. Any other questions? Feel free to put them in the chat, or you can also unmute and ask if you'd like.
Okay. You also do model calibration modeling based on the experiments. Yes, of course, because we have to do a lot of parameterization just to make sure the model results fit the data. And actually, it's a slow process because you cannot do auto fitting or optimization pretty much automatically with this case. Each runs take at least a day, and sometimes you have, because of stochastic simulation, sometimes you have to do multiple times to get some consistent results, and it is time consuming. Yeah. Other questions? because there's a lot of parameters to consider. I'm wondering which parameter did you alter and consider? Okay. Um, I okay. There are um, parameters, uh, both the parameters for the cellular behavior as well as parameters for the gene network were varied. All right. So the and the yeah, so you have to basically um, vary or calibrating all those parameters to be able to match your model behavior with the data. Um, I cannot pinpoint exactly one parameter or not. But to give you an example, since the doubling time of the B cell in this case about 5 hours, right? So definitely you can define your cell volume growth parameters based on that, which is right a natural log 2 divided by the 5 hour will give you the K value for that cell volume growth. Okay. Um, yeah. And it takes a lot of trial and errors to get it right, I'll say. Mhm. Yeah, yes, I just said that trial and error process. Um, yeah, I didn't use any optimization. It just um, I, I don't think it's feasible in this case.
Yeah, we'll this question of parameterization and how to select, the best values that fit your model is a kind of one of the core questions of computational modeling in general. We'll actually have a couple of sessions on that next week during the advanced materials on how to select parameters and then how to optimize for different parameter sets. So yeah,
Right, but
good question.
yeah, but the general question hitting is like for simulation like this is very slow, take a day or something to finish,
Mhm.
right? I mean, it's gonna take forever, uh-huh, to optimize, basically.
Right.
Uh,
Well,
yeah,
that'll combine well with our session this year on running CompuCell on a supercomputing cluster. Because then you can run, 100 different runs at
right.
once,
Actually,
essentially.
I was already running this with our, no, actually, no, I did this on my laptop. Yes, I didn't do this with our class or anything. Um, right. Yeah, yeah. So great paper, by the way. Thank you. Yes, because just for modularization for a simple OD model is not trivial. Then how about this agent-based model? I'm wondering how to do that. Yeah, I'm learning that as well. Definitely. Hmm. Yeah.
Yeah. Yeah, Osmond, we should continue this discussion in the future because it is a big question.
Hmm.
And there's not necessarily like a correct answer. It's sort of
Hmm.
method dependent. So yeah, we'll continue
Yeah.
discussing that. So, okay,
All
well, I
right.
think we're at the end of our session here. Thank you so much, Dr. Zhang, for coming and presenting for us. Um, I know people had a little bit of issue getting the CompuCell code to run, and then there's with the Python visualization, I know you probably need to install a couple of packages that aren't standard, so I'll put together a how-to document for anybody who attended and who's interested in running the
Hmm.
demo code. You just have to install a couple of visualization packages into your Python installation. That's not difficult. So um,
All right.
we'll get that
Yeah.
put together and sent out. And thank you everybody for coming today. So for those of you who are taking their lunch now, please remember to rejoin the main room at 2:30. No, 3 p.m. I'll send a reminder email. 3 p.m. Yes, Nashim, thank you.
Yeah.
Um,
All right. Bye.
bye
Yeah.
everybody.